Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Rathangadhara Nammalwar from Sartorius.
Thank you very much for the kind introduction, Leah, and welcome everyone to today's Ask the Expert session, in which I will be talking about Sartorius's optimized expression systems for increased productivity in CHO DG44 based cell line. So going on to the agenda. So I will start with an introduction to Sartorius, and then we'll focus on the cell line development division here in the South German city of Ulm. Then I will move on to a few of the salient features of how an optimal cell line development platform should look like. And then I will also talk a little bit about the cell line development platform of Sartorius. And finally, I will also present some data which shows the importance of having an optimal expression vector in order to achieve enhanced productivity in a CHO-based cell line development platform. So let's go ahead. So yeah, Sartorius is an international partner for life sciences research as well as biopharma industry. It's a multinational company, and we are located in 60+ locations worldwide. And the headquarters is here in Göttingen in Germany. And as of December 2020, we were about 10,500 people with a worldwide sales revenue of about 2.3 billion euros. And now, as I mentioned before, our facility here is more specialized on the cell line development, media development, as well as upstream process development. We are located in the southern part of Germany in a city called Ulm. We are about 130 employees here on site. And as I mentioned before, our expertise goes in the direction of CHO-based cell line development, media development, as well as process development. And in our facility, we have the capabilities of generating about 100 cell lines per year. And the teams are also having very specific portfolios in a way that there is a targeted team which works on the product development, meaning to say we are constantly working on generating new products and services for our clients. And on the other hand, there are also focused teams which are working exclusively on the client projects. So with this kind of scientific as well as technical expertise, our clients get the luxury of having the perfect infrastructure to get their cell lines expressed. Okay, so as I said before, I have put together a few points which marks a vital or a very ideal cell line development platform. And here are those. So first of all, high productivity, which is a very absolutely important goal without increasing the cost of goods. The next is short CLD timelines, and CLD in this case, I mean from the stage where you have your DNA until you reach your research cell bank, the timeline that is required should be reasonably short. And another very important point is that the whole process should be preferably antibiotic-free, meaning to say that the whole process should be able to be done without components such as MSX and MTX. As far as the host cell line itself is concerned, it is highly desirable that the host cell line has full traceability. And another very important point is the scalability. So it's important that the processes that are developed are scalable in order to avoid extended media and process optimization times. And last but not the least, the technology should also be able to produce a product which has an optimal product quality in order to ensure the required PK and PD profiles. So keeping that in mind, how does the Sartorius cell line development platform look like? So that's the entire, let's say, process of the cell line development that we have here. So it starts with the process of gene synthesis, for which we have an external service provider from whom we get the genes synthesized. Once the genes are synthesized, they are integrated into Sartorius's proprietary expression vector by cloning, and this expression vector is then transfected into our CHO DG44 host cell lines, and pools are generated. Pools are expanded, and pool analytics is gemacht. And at the same time, you also lay out single cell clones. These single cell clones are also expanded, and then pool analytics are done, or clone analytics are done also on the single cell clone stage. And finally, the top performing clones are also laid out as the so-called research cell banks. So this whole journey from gene synthesis until the research cell bank, in our case, takes roughly about 14 weeks. And in Sartorius, we believe that an efficient cell line development platform relies on 4 major pillars: the host cell line itself, so CHO DG44, our host cell line, the media, the upstream process design, and also the expression vector. So with Sartorius, our clients have a very reliable CHO DG44 host cell line. So it, as we all know, is DHFR deficient. It is adapted to suspension growth in chemically defined medium. It has a fairly low doubling time. The host cell itself has a doubling time of about 18 hours, and the production clones have a doubling time of about 16 hours. When it comes to media, we have a dedicated team which is working constantly on optimizing our media, so our selection media as well as our production media, according to our processes and our host cell line. So that's also an added advantage that we have on our platform. The next aspect is the upstream process design, and here there are also dedicated teams which are working on standardizing the processes with the defined media and the setup so that they lead to robustness and scalability, so that there is an easy transfer to a manufacturer or CMO. Last but not the least, the 4th pillar of an efficient CLD technology is the expression vector. And the expression vector that is present on the Sartorius cell line development platform is a result of years of research and development. So our product development teams have worked intensively in adding optimized genetic elements in order to ensure that there is a stable and efficient expression of the product. And as part of this talk also, I have chosen 2 of those new elements which we have tested on our expression vector, which has shown significant increase in the performance of our cell line development platform. And to that data, I will come in the subsequent slides. Before that, a quick look at the track record that we have so far with our CHO cell line. So we are proud to say that we have generated more than 100 cell lines so far. And when you look at the modalities, it covers the entire spectrum of the different therapeutic modalities. And when we look at the titers, we see that across all these modalities, we are able to reach high titers of 5 to 8 grams per liter very often. So that's so far about the Sartorius cell line development platform. And in this talk, as I mentioned before, I would like to talk a little more about the expression vector itself. As I mentioned before, this is an expression vector which is a result of R&D of several years, and we have optimized this in a way that we have the optimal signal matrix attachment regions, optimal promoters, and other elements. And the topic of today's talk, what I've chosen is 2 other elements which we have tested in the recent past, which are the 5' and 3' untranslated regions, also known as UTRs, and signal peptides or SPs. And untranslated regions, as we all know, are very crucial genetic elements which are very important in directing a transcribed mRNA to the right cellular compartments, and it also has, or they also play a very important role in mRNA stability of the transcribed mRNA. Signal peptides, on the other side, are working on the protein level, that they also are very crucial in driving the translated protein to the right cellular compartment so that they are very efficiently also secreted later on. And just as an initial view, what I want to say is at the end of this study, what we were able to achieve is 2 optimized expression cassettes on our cell line development platform, one optimized for a single chain protein such as Fc fusions, etc., and another set of UTRs in signal peptides, which are optimized for the expression of multi-chain proteins, typically like an IgG type molecule. And so our clients also have the advantage of choosing between these two, depending on the type of modality they come to us with. So now I will just tell a little bit about how we went about doing that. So in order to test these untranslated regions as well as signal peptides, we collaborated with an external partner or a company called as Unitargeting Research. So this is a company which is located in Norway, and their expertise is in the area of designing such genetic elements which have shown to have a very positive effect on productivity on different production cell lines. So basically what we did was we contacted Unitargeting Research, and they designed for us several 5' UTRs, several 3' UTRs, and several signal peptides. I will go to the details of it in the next chapter or the next slide, but what we did then was we cloned in these different genetic elements onto our then standard expression vector. And what we did then was we generated these so-called large pools, or LPs. So these are basically you transfect in 6 well dishes, and then you expand them until shake flasks, and also perform your selection through the process. And when the selected pools reach the shake flask, you then inoculate a 14-day fed batch procedure, and then you look for the titers at the end of this procedure, how it looked like. So this gives us a very fast way of screening for different elements. And what we see here is the results of that initial screening experiment. So as I mentioned before, these are the different combinations of 5' and 3' UTRs, which were given by Unitargeting Research. So on the left-hand side, I have all the different combinations which were provided by them for double-chain proteins. And on the right-hand side, there are those combinations for the single-chain proteins. And in summary, what I want to tell is we had in total 3 different 5' UTRs, 3 different 3' UTRs, 3 different signal peptides, and a poly A in total that we had tested in different combinations. And here is a result of that initial screening. So these are pool fed batch titers in comparison to the standard. So the standard was set to 0, and what we see is that indeed we have one combination each of those UTRs in case of antibody, as well as with a single chain product such as an FC fusion, which seems to have shown an increase in productivity in the first view. So the obvious step for us was to check or to validate this with more products. So what we did was we expanded that further, and we did the same exercise once again with 3 other products. So 3 antibody molecules and 3 FC fusion proteins. And these are again the results. These are the final pool fed batch results from those pools. And what we see here is always the standard is shown in black. The UTR beta tech, which is the technology of the untranslated regions, we see in blue. And the yellow bars represent the combination where we have put the UTRs as well as the signal peptide together. And what we see clearly is that there is a significant increase in productivity between our then standard vector and quote unquote the new expression vector, which contained the optimized genetic elements. So that holds true for antibody molecules as well as FC fusion molecules. So this was all the story from the pool level. So the obvious next step for us was to look at the whole thing at the clone level. And what we did there was we repeated the exercise of transfecting the cells with expression vectors, generating the pools, laying out single cell clones using a FACS device, and then expanding the clones and performing clone evaluation either in a shake flask fed batch like we did in the previous round, or we also performed clone screening using our AMBER 15 systems. And how does that look like? So these are now 3 different monoclonal antibodies and 2 different FC fusion proteins, what we tested there, and it's always the then standard expression vector against the new vector. And we see that we here in this case, we use the top 12 clones, in this case, the top 10 clones. And what we see is that the leading clones or the lead clone itself has a very significant improvement in terms of productivity with all the different products. So that way, this shows that the technology really worked for us, and we were able to improve our productivities with the quote-unquote new expression vector. And as a last and final step, what we then did was we took the whole thing and scaled it up, and to see if it holds good also in case of bioreactor setup. And here again, these are the same monoclonal antibody 1, 2, and 3. And in yellow, it's the amber curves, and in black, you see the bioreactor curves. And what we see is that in these two cases, it's very much comparable. Here, in this case, the growth curves go a little bit away from each other, but I have to point out that this was a comparison between a shake flask versus a 5-liter vessel. So I think due to the optimized conditions in a bioreactor, you can expect that this was a little bit better, but nevertheless, when you look at the titers, what we see is that it's very much scalable and very much comparable, and these elements really seem to have a very significant effect on the productivity itself. So that was basically what I had to tell today, and I hope with all this data, I was able to convince you a bit that having the right genetic elements on your expression vector is very important, and it is also a possibility to improve your cell line development platform to have or achieve higher productivities. So I thank you very much for your attention, and I'm happy to take any questions.
Okay, great. So do you think it is important to have different UTRs for single chain and double chain genes of interest?
Yes, at least this is what we have noticed with the experiments that we have done, right? So we did have the same UTRs between the double chain and the single chain proteins, but this did not seem to yield equal benefits. So that's the reason why we had to go another round back to unit targeting research to find out if this could be changed, and then they suggested us other specific UTRs which are specific for single chain proteins, and this really at the end did the trick. So it seems really that it is also important to see what kind of product you're dealing with, and based on that also decide which UTRs you want to use.
Okay. And how important is the combination of signal peptide with the UTRs?
Yes, that's a good question, and as we have seen in the data before, only having the UTRs does not necessarily always bring the full benefit, but it seems that in our hands at least, that the combination of the UTRs with the signal peptides was the better option. So I would say yes, it is important to have both.
Okay. And do you have any readily available stable cell lines that can be used for research purposes that produce IgG4 or Fc fusion protein?
Sorry, I didn't get the question.
Do you have any readily available stable cell lines that can be used for research purposes that produce IgG4 or an Fc fusion protein?
Yes, we do. So we do have cell banks from the clones that we have generated. So we do have such cell lines available, but I do not know what the background of that question is. In a way, if we could source this out to an external partner, that is something where I do not know if there are other aspects which would be involved. So we could get back to the person on that.
Okay, great. So it looks like we have time for just one more question. If we didn't get to your question, we will be passing them on to Dr. Nammalwar, and he can follow up with you directly. So the last question for the webcast is the new expression vector already part of the CLD offering at Sartorius?
So yes, it is part of the CLD offering. So we do have projects running with the latest version of the vector.
Okay, thank you. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.